Hi there, my name is Dr. Stewart, and today we're going to talk about this novel coronavirus, COVID-19, and we're going to look at its structure, and we're going to look at how this virus is able to infect a host cell, what it does inside the host cell, and how it's able to replicate inside of one of our cells. So COVID-19 is an example of a coronavirus, and you've probably heard this before, but coronaviruses have been around a long time, and they are common causes of respiratory illnesses. In fact, like 5 to 10% of all acute respiratory illnesses are caused by these coronaviruses. They're small, they're spherical in shape, and they have these little glycoproteins called spikes that emerge from their envelope that gives them this kind of name of a coronavirus, like a crown. And there's been a couple of different outbreaks over the years. Recently we had SARS, the outbreak in the early 2000s. That accounted for about 700 to 800 deaths. And then in 2012 and 2014 and 15, we had the MERS outbreak, the Middle Eastern Respiratory Syndrome, which accounted for about 900 deaths. But this recent novel coronavirus, COVID-19, is much more of a problem, obviously. Okay, so let's look and see what this guy looks like. Now, size-wise, he's really small. He's only about 60 to 140 nanometers in diameter. And if we go back to our original little size chart that we looked at here from a couple lectures ago, here's a red blood cell. It's about 8 micrometers. Here's E. coli, about 3 micrometers in length. This is the smallpox virus, which is about 0.3 micrometers or 300 nanometers. So that means that the coronavirus is going to be about, I don't know, half the size of this guy. So the coronavirus would be about that big. So that would be COVID-19 right there. And his diameter is about 0.1 micrometers or 100 nanometers. So 0.1 micrometers. So he's very small. Now, if we were to zoom in and look at what this guy actually looks like, he is an enveloped virus, which means he has a protective cell membrane that covers him. That's what I'll draw. That's what I'm drawing kind of here in purple. So this purple kind of ring represents the envelope or the cell membrane of this COVID-19 virus. Now remember, a cell membrane is primarily composed of phospholipids, which it has stolen from its host cell. So that's what all these little lines through the cell membrane represent. Those are all the fatty acid tails of this phospholipid bilayer. I'm really simplifying things. It looks like there's only one layer of these phospholipids, but there's really two. But that's what these hatch marks represent, are all of the phospholipids that make up the cell membrane. So we'll talk about the inner constituents of the cell membrane here in a little bit, but inside the envelope or the cell membrane, that's where you're going to find the viral genome that is covered in its capsid. So this green kind of color, that represents the capsid, and the viral genome is going to be located inside this capsid. The coronavirus, or COVID-19, let me label him right here. So this is COVID-19. He is going to contain what's called a nucleocapsid. And really all that's going on here is this viral genome in black is going to be kind of encased in these proteins that protect it, that create the capsid. So they're really the genome and the capsid are kind of intertwined. So in black we have the genome right here. And then this green kind of color, that's the capsid. That's going to be located right there. Now together, those two guys create the nucleocapsid. Great. Now let's talk about what we're going to find inside that envelope. So the envelope is this starts with this purple ring-like structure. The first thing that you're going to find there are a series of glycoproteins that emerge like spikes. They're called spikes. They emerge out of the envelope. And these guys, they're called spikes. and they're going to kind of look like this. So we're going to draw all these blue kind of spike-like proteins, which are projecting out of the envelope, out of this membrane, and that really is what gives coronavirus its name, because it looks like a bunch of spikes on a crown or something. All right, draw a couple more. Now, what's important about these spikes is these spikes are the ones that are actually going to bind to special receptors in the cell membrane of our hosts, of our cells, of the host cell. So these guys are spikes, and they're made up of glycoproteins. Now, another thing that you're going to find inside the cell membrane are going to be membrane proteins. And these membrane proteins are really important with the assembly of the virus inside the host cell. So these membrane proteins, I'm going to draw these guys in red. So they're just going to be little proteins that are hanging out inside the cell membrane like this. And they're really important with how the virus is assembled inside the host during replication. So this is a membrane protein. Right there in red. And then the last protein that we're going to talk about is called an envelope protein. I'm going to draw this guy in this little green color. There's not many of these envelope proteins, but they're also kind of stuck inside the cell membrane. And they are really important in how the virus interacts with the host. And they're also, with the host cell, and they're also important with assembly as well. So this is in green, you're going to have the envelope protein right there. And everything else that's in purple, those are phospholipids that just make up the envelope, that cell membrane that makes up the envelope. Okay, good. And it's worth mentioning these spikes. These are really what binds to proteins in the cell membrane of the host. And the specific protein that it's going to bind to, these guys are called angiotensin converting enzyme 2. Okay, so they're abbreviated ACE2. Now, what we're going to do is we're going to zoom out and we're going to draw the single virion of COVID-19, and we're going to follow it from the time it binds onto the host cell, how it enters the host cell, how it replicates its genome, how it produces these viral proteins, and how a new virion is produced. So what we'll see here is we're going to kind of zoom out a little bit, and then I'm going to show you that this pink line, that represents the cell membrane of one of our host cells. Like this could be a cell in our lung, right? This could be a lung cell. And okay, so here we have the cell membrane of one of our host cells. And the cell membrane of one of these host cells is going to contain the specific protein marker that COVID-19 is attracted to, and that's called the angiotensin converting enzyme 2. I'm going to draw these guys in this dark blue kind of color. So we have these angiotensin converting enzyme 2, and it's just a special protein marker in the cell membrane that this coronavirus is going to be attracted to, and it's going to bind to it. So this is ACE2, which corresponds to angiotensin converting enzyme number 2. And that is the marker that these spike proteins are attracted to. So up here, we're going to have a molecule 1 virion of the coronavirus. Draw him out. We're going to have his envelope in purple. I'm going to simplify things a little bit. We're going to have the genome on the inside. To simplify things, I'm not going to draw all these components. I'm just going to stick with the spikes. So we're going to have these spikes that emerge out like this. Well, these spikes are super attracted to this ACE2 protein in our cell membranes. Like cells in our lungs are going to be loaded with these receptors or these proteins. And so COVID-19 is going to be really attracted to those. Now, one thing that's important is that when you compare COVID-19 to other coronaviruses like SARS that have caused respiratory illness, the spikes of COVID-19 are like 20 times more attracted to this ACE2 protein than the spikes of like older coronaviruses. So that could explain why it is so effective at infecting people and is spreading from person to person. So what will happen is that this coronavirus, this virion, is going to bind onto the ACE2 proteins. Let me just redraw them right here. We have the envelope, and we got the genome on the inside of that capsid. And you'll see that those spikes are really stuck to and binding to the ACE2 proteins in our cell membrane. What that's going to do is it's going to trigger endocytosis. That's how this coronavirus gets into our cells. It's going to trigger endocytosis so that the cell membrane of our cells is going to engulf this virion of COVID-19, and it's going to create this little vesicle inside of our cells that contains the virion of the virus of COVID-19. So we're going to have this little vesicle here. That's literally like a piece of our original cell membrane. Inside that vesicle is going to be that single virion of COVID-19 that has been engulfed. Now, just for simplicity and for just to be complete, actually, let me draw all the spikes of COVID-19 in here. And then we're going to have all of those ACE2 proteins in the original cell membrane. And they have bound together. Now, it's worth mentioning now that, remember these envelope proteins? One of their roles is to trigger this endocytosis. They really help with the interaction of our cell membrane with the virion of the COVID-19. Oh, wrong color. Okay. Now, once this vesicle has entered into our cell, the envelope of the COVID-19 is going to fuse to the outer vesicle of this vesicle, and that's going to release the inner capsid and genome into the cytoplasm. So these guys are, this inner membrane of the envelope of the virus is going to fuse with the outer membrane of the vesicle, and that's going to create this type of situation to occur. So they fuse, which is going to release the capsid into the cytoplasm. Now, remember that the inner kind of part, that contains all these spikes of the virus, and the outer part, that's part of our cell membrane, so that contains all these ACE2 receptors, just so we can keep track with what's what. Now, the genome, the viral genome, is now going to be released. The viral genome is now going to be released into the cytoplasm. But if you remember, it's coated with this green capsid, so another process needs to occur, and that's called uncoding. So the proteins associated with this capsid in green are going to be removed from the viral genome, so the viral genome is then exposed to the cytoplasm. And the first place that this viral genome is going to go, it's going to go actually to a ribosome of one of our hosts that exists inside our cell. So this is one of our ribosomes. Ribosome. This is one of the ribosomes that we've made inside of our cells that was doing the job of, our cell up until now. Now, quick review of the job of a ribosome. A ribosome has the job of taking our genetic code as a guide, and according to that genetic code, it's going to make proteins that that cell needs to make in order to do the jobs of that cell. Okay. In this case, this virus has given the ribosome some viral proteins, so it's not going to make the proteins it should. It's going to start making viral proteins. And the first protein that it makes is this really special and important protein when it comes to the virus, and that is called the replication and transcription complex. So I'm going to draw this guy in orange, actually. I don't like to. I'm going to draw him in orange. So in orange, we're going to have this replication and transcription complex. Now, this is a viral protein that was made from our ribosome using the viral genome as a guide. So this is the RTC, which stands for replication and transcription complex, RTC. Now what this guy does is that he's going to take other parts of this viral genome that have been encoded. So we're going to have other parts of this viral genome that are going to come over here to this replication and transcription process. And what this guy does is he starts to really crank out more copies of the viral genome that can be then interpreted by other ribosomes inside of our own host cell. So this guy just starts making copies and copies and copies of the viral genome, and it's going to make them in the form in which these host ribosomes can understand. Now it's worth mentioning this viral genome comes in the form of positive single-stranded RNA. That's the form of this viral genome. This replication transcription complex is going to create new forms of this single-stranded RNA genome, but each little piece of this replicated genome is going to encode for a specific part of this virus. Like one piece of the code that it produces is going to encode for the genome itself. The other part's going to encode for the spikes, another part's going to encode for the envelope proteins, and another one's going to encode for the membrane proteins. So to kind of keep the color code consistent, we're going to have this replication and transcription process. It's going to produce some genome that corresponds to more viral genome. So this is to the viral genome in black. It's going to produce some other genetic code that is going to produce the spikes. So we'll do that in this like little blue color because that's what color the spikes are. All right, so that's for the spikes. Then it's going to encode stuff for the membrane proteins. Which color did I use for them? Oh, orange, I think. Yeah, I used orange. So then we're going to have the membrane protein. And then finally in green we'll have the envelope, the RNA that encodes for the envelope protein in green. So this is envelope. Now, just to put this into perspective, this is not the actual proteins. These are just different versions of RNA. So RNA that encode for each piece of the virus that needs to be produced to produce more viruses. Now what will happen is all three of these types of RNA, the ones that encode for the spikes, the membrane, and the envelope, they're going to go to the endoplasmic reticulum. Specifically, they're going to go to ribosomes that exist in the endoplasmic reticulum. Now remember, that's a part of our cell that contains a lot of ribosomes that its job is to produce proteins. Well, it's going to use this RNA as a guide to produce these very same proteins. And these proteins are going to be placed right here in the membrane of the endoplasmic reticulum. So you're going to have some membrane proteins here, right? The ribosomes are just cranking them out and putting them right next door. You're going to have some envelope proteins that are being produced. We'll draw these guys in green. It's looking very similar to the envelope of COVID-19 all of a sudden. And then it's going to produce a lot of these spikes, just like before. Now, the viral genome that is going to go inside the middle of the new viral particle, it actually is going to be coated with capsid in the cytoplasm. of the cell itself, so that doesn't happen in the endoplasmic reticulum. It's going to happen right in here, and so you're going to get this new kind of viral genome, which has been replicated from that RTC complex. It's going to come over here in the cytoplasm where it gets coated with, let me see if I can draw this really quick, I forgot to do the green. It's going to be coated in green with its capsid. Now what will happen is that all of these new proteins that have been produced, they're then going to be sent to the Golgi apparatus. Remember, the Golgi apparatus is a part of the cell, it's an organelle that's responsible for packaging and moving new proteins. Well, that's exactly what it's going to do. It takes all these newly made viral proteins and it's going to package them and produce a new viral particle. The Golgi apparatus is really what produces the assembly process in replication. So now what you're going to have is you're going to have all these spikes which have migrated out here. They're now going to be located in this little vesicle, this little piece of the Golgi apparatus. You're going to have all these membrane proteins, right? And you're even going to have these oh, I hope this was all included. And you're even going to have these envelope proteins. So if this wasn't even really that visible, you have all these bits of RNA which get sent to the endoplasmic reticulum. Our own host ribosomes are going to produce new viral proteins in the cell membrane of the endoplasmic reticulum. These viral proteins, such as the spikes, the membrane proteins, and the envelope proteins, get sent out to the Golgi apparatus, where the Golgi apparatus is literally going to assemble a viral particle by taking this viral DNA, which has been put into a capsid. It's going to engulf that capsid, and then all of a sudden we've got a new little viral particle, complete with its viral genome inside the green capsid, and now it is located inside of a vesicle. That vesicle is sent. The vesicle was made from the membrane of the Golgi apparatus. You can see how it kind of just folded in on itself. And now we're going to have all these little viral proteins like that. You're going to have the spikes coming out like this. And then finally, what will happen is that through exocytosis, this vesicle fuses with the outer cell membrane of our host cell, and a new viral particle emerges. So we started, the virus came in, new viral particles came out. I drew one coming in, one coming out. It obviously doesn't work like that. You're going to have hundreds and hundreds coming out. And that's typically how the life cycle of COVID-19 progresses. I think in a nutshell, the main take-home messages of this are that the spikes of the virion are what bind onto the ACE2 proteins of the host cell that triggers endocytosis, creating a vesicle that contains the viral particle. The envelope of the virus and the vesicle fuse that releases the viral genome and its capsid into the cytoplasm. The viral genome is uncoded, so the capsid is lost. This viral genome then goes out to a host ribosome where it produces a bunch of replication and transcription complexes. This is RTC, and that is these guys are what is going to replicate the viral genome many times over, so that you're going to have little bits of RNA that encode for the envelope protein, the membrane protein, and the spikes. Also the viral genome. The viral genome is going to be coded in the capsid inside the cytoplasm, whereas the RNA for the spikes, membrane, and the envelope, they go to the endoplasmic reticulum, whereas the ribosomes actually create these viral proteins. These new viral proteins are sent to the Golgi apparatus. The Golgi apparatus puts everything together into a new viral particle. It emerges out of the cell through exocytosis, and now we have new virion of COVID-19. right here. So you might be wondering, like, well, how can we stop this process from occurring? Well, a couple of different ways, potentially. You can interfere with its ability to bind onto host cells. That's a really good potential. You could interfere with its ability to replicate its genome. That would also prevent the virus from from replicating. Or you could prevent the Golgi apparatus from effectively assembling the different pieces, among other ways. It's worth mentioning that after the endoplasmic reticulum cranks out so many virus particles, it kind of wears itself out, and that's what leads to the death of the cell, is that this endoplasmic reticulum really just gets overworked by producing all these viral particles from the viral genome that's now filled up with the cell. That's about it. Thank you.